So far with the immune system, I've talked a good bit about antibodies, but I haven't yet told you exactly what antibodies are. So we're going to fix that, all right? We're going to talk about what antibodies are, all right? Antibodies are also called immunoglobulins, all right? And in terms of those immunoglobulins, a lot of times you'll see that abbreviated capital I, small g, all right? I may abbreviate antibodies as ABS, okay? Just so you know what I'm referring to. But in terms of antibodies, or those immunoglobulins, this now is going to be part of our humoral response. And essentially what those antibodies are, these are going to be proteins, all right? And in terms of those proteins, they're going to have some regions associated with the antibody that are going to have particular functions, all right? And in terms of that, I'm going to kind of draw this on the board here in parts. Oop, I want that to go the other way. And the thing about antibodies are that they're made and then assembled together, all right? And where they're made is going to be within the B cells. They're also going to be assembled together within the B cells. All right. And in terms of that, if you notice, I've got two different colors here. The blue part is going to be the heavy chain. The yellowy green is going to be the light chain. Basically where that comes into is size, is the heavy chain is bigger, the light chain is smaller, all right? And in terms of the light chain, all right, that's going to create a region with the heavy chain. That is known as the FAB. If you want to call it FAB, that's wonderful, okay? In terms of that FAB region, this is going to be where our antigen, or that PAMP on the outside of our invading pathogen, is going to bind to our antibody. This is going to be the part that recognizes it, all right? So in terms of that, this is going to be the highly specific portion of the antibody. All right, so the FAB portion is where the antigen binds, all right? You've also got the region in blue here, which is known as FC, all right? A lot of times you'll see this referred to as a constant region, all right? But in terms of this constant region now, I'm going to kind of put a little blip in green over here, and a little blip in green over here. This is going to be the portion of the antibody that complement will bind to, all right? So in terms of our antibody, this is going to be generally how things are arranged, all right? There can also be another site down here. Let's make these kind of as orange footprints, because some antibodies are actually going to be anchored to cells, okay? So in terms of that, this is going to be our binding site for cells, okay? And usually what makes this up are some glycoproteins, some carbohydrates, all right? Not all antibodies are going to have a region that they're going to attach to a cell. Some antibodies are going to be free-floating, all right? But some do have the ability to bind to specific immune cells, all right? So in terms of our antibody composition, all right, essentially what we've got here, we've got 4 protein chains, all right? Our protein chains, we've got 1, 2, 3, 4, all right? That's just to show you that we've got 4 individual proteins there, all right? And those protein chains are going to be held together by disulfide bonds. All right? So in terms of those antibodies, the antibodies that you have circulating are assembled within the B cell, all right, and released as the mature antibody that I have drawn up here, all right? But this is another reason why fever can be dangerous, high fevers, because those disulfide bonds are going to be heat sensitive. And if you break those disulfide bonds, our antibody falls apart, and now we have 4 proteins that are just floating around, and an antibody doesn't work unless all 4 chains are assembled together, okay? So in terms of that, our heavy chain and light chain are going to be held together by disulfide bonds, all right? And in terms of that, there are a couple more regions I want you to know that are associated with our antibodies that are going to become important. And I'm going to erase the numbers here because I need to draw in those other regions there, okay? So in terms of our antibodies, you are going to have some constant regions, okay, like those, and then I can use, let's use orange, and then the outermost regions are referred to as the variable regions in our antibody. Okay? And the fact that these are variable is what allows us to produce different types of antibodies. Our body is capable of producing hundreds and thousands of different antibodies specific to a particular antigen, all right? And it's because we have those constant regions and those variable regions that allow that to happen, okay? And in terms of our overall antibodies, these are the structures I want you to know on the antibody. I'm going to go over a little bit of detail as far as some of the specific immunoglobulins that are circulating within your body and what they do, and I want you to be familiar with that. But I want you to know the basic parts, the anatomy essentially of the antibody, the fact that it's held together by by disulfide bonds, all right? And also with those antibodies, this is going to be where our complement's going to bind, all right? If you have any questions, please let me know.